Hello, all. Good morning. Today, we are going to start a topic which is very interesting. I like the topic very most. It's plant tissue culture, PTC in short. Actually, it's a lengthy topic, so that we could not see it in a single presentation. So, I had split it into 5 to 6 presentations. So in today's presentation, we will see just about its introduction and various types of PTC. So plant tissue culture, now what it is, if you cut the word plant tissue culture into two terms, plant tissue plus culture. Here culturing means growing, developing or producing. Right. So what we are going to produce? So in plant tissue culture, what we are going to produce, what is the final product is any one of two, which are either callus or a new plant. So we are going to produce a product called as callus or else a new plant, right? From where we are going to. So the product is either callus or new plant. Static material, it's plant tissue. So we are going to use a single plant cell or else a group of cell, which means tissue. So we are going to use a single cell or a tissue, and we are going to produce it into a new plant or as a callus, which is called plant tissue culture in short, right? So it is an in vitro cultivation of a plant cell or tissue into a callus or a new plant by using artificial nutrient medium under aseptic conditions. So the entire procedure what we are going to do here will be done under aseptic conditions, right? First one. Number two, it's an in vitro technique. So we are going to do all these procedures in a lab, right? So it's an in vitro technique in which we are going to use a plant cell or else a tissue, and we are going to develop that cell or tissue into a callus or a new plant by using artificial nutrient medium. So we are going to take a plant cell, we are going to keep it in a nutrient medium which are done under aseptic condition, so that the cell will get converts into a callus or else as a new plant. Fine. So types of PTC. So here we are categorizing it into two categories. One, we are classifying based on the type of H plant we are using. Number two, based on the methodology. Right. So based on type of H plant. What is meant by H plant? H plant means whatever the cell or tissue we are going to use to produce a new plant, that particular cell or tissue will be called as a H plant, right? So the plant cell or else a plant tissue what we are using to develop a new plant will be called as H plant. So based on the type of H plant, PTC may be root tip culture, leaf primordial culture, shoot tip culture, anther and ponal culture, flower culture and so on. So based on the H plant, we are categorizing it into numerous categories. So if you are going to use root tip, cells present in the root tip, right, then that particular culture will be called as root tip culture, right? So we are going to isolate a cell from the root tip that will be inoculated in a culture medium to develop a new plant. Fine. If you are going to isolate a cell from a leaf and we are going to use it, then that is leaf culture, shoot culture, flower culture, so on. It goes on, right? So based on the type of H plant, there are numerous types. Fine. Next category, based on methodology. So what's a procedure we are going to follow to convert that single cell into a plant? Based on the procedure, PTC can be broadly categorized into three categories. One is callus culture, Number 2 is cell suspension culture. Number 3 is protoplast culture. All are nearly same with minor changes. Alright. For example, callus culture. As the name indicates, here we are going to produce the callus. The final product what we are going to produce is a callus. So we are going to use a single cell or a tissue. We are going to inoculate in a medium. We are going to wait for certain period of time. After which we are going to remove the callus what is formed and using it for further purposes, which is called callus culture. Fine. Number 2, cell suspension culture. So, in callus cultures, generally we will be using a solidified nutrient medium. Solid nutrient medium. Okay. If you are using a liquid nutrient medium, then that will be coming under cell suspension culture. So in cell suspension culture, you will be using only liquid nutrient medium. You won't use solid medium. Fine. Number 3, protoplast culture. What's meant by protoplast? A cell without cell wall is called protoplast, right? So here the explant what we are going to use is not a cell, it's a protoplast. So if you are using a protoplast as a explant, then it is protoplast culture. Fine. So everything, the procedure will be nearly the same except minor changes. In protoplast culture, you will be using only protoplast as explant. In callus culture, you will be using any cell or tissue, but final product will be a callus. In cell suspension culture, you may use any explant, but the nutrient medium will be liquid, right? So that's about the introduction. So next topic we'll see in the next class. Thank you.